It's now a great pleasure to welcome live from Foundation Fighting Blindness in the USA, Ben Shaberman. Ben leads the Foundation's professional outreach team, educating eye care professionals about resources for people with regard to inherited retinal diseases. In addition, he writes stories on science and research for all of Foundation Fighting Blindness's publications, and he also presents the latest advancements in retinal research. So it is a great honor to welcome Ben Shaberman live from East Coast of America.
Well, thank you, Neil. It's always an honor and a privilege to join Fighting Blindness Ireland's Patient Engagement Day. And actually, the picture I have on this slide on the left is of me visiting Cliffs of Moher when I was in Ireland about 3 years ago, and I came to Patient Engagement Day live. That was a great meeting, and I hope to come back to Dublin in the not too distant future. I will be providing an overview of many different types of projects that are either in clinical trials or getting close to clinical trials, at least most of them. And it's a very exciting time with so many clinical trials underway. We count somewhere in the neighborhood of 45 trials for emerging therapies. And because of time, I can't cover them all, but I covered some of the projects that I think are most interesting and furthest along in development. Just a little bit about the Foundation Fighting Blindness. There we go. We were founded, we're celebrating an anniversary as well. We're celebrating our 50th anniversary. Like Fighting Blindness Ireland, we were founded by a small group of families who were trying to find answers to their retinal conditions at a time back in the early 70s when little was not only known about the retinal diseases, but even the retina as well. And we've emerged to become a leader in driving research for these conditions. We've raised more than $800 million toward our mission. We fund a lot of different types of research, obviously development of treatments and cures. We fund natural history studies to understand disease progression. We fund research projects to move therapies out of labs into clinical trials. We're even funding some trials. And we also have what we call our RD fund. It's a venture philanthropy fund where we are co-investing with venture capitalists to get therapies that are either on the doorstep of a clinical trial or in an early stage clinical trial, to get them to the point where we can attract outside investments from big pharma and big biotechs, because clinical trials cost lots of money, especially the later stages, easily tens of millions of dollars and often hundreds of millions of dollars. And of course, those dollars are US dollars. That's the metric I use. So to start off, I wanted to give a brief overview of LUXTURNA, and I really appreciated the compelling and inspiring testimony we heard from Jake earlier. LUXTURNA has really been, as we say, a game changer in the world of gene therapy. So LUXTURNA, as I think you learned, is for people with mutations in the gene RPE65. Mutations in that gene cause a form of LCA, and in some cases, a form of retinitis pigmentosa. And this is another good reason to get genetically tested, so you can figure out if you have RPE65 or another gene that might qualify you for an emerging therapy. So the research for LUXTURNA really began in the mid-90s. Canine studies in the late 1990s, early 2000s were a great proof of concept. There's actually a naturally occurring dog model of the condition that LUXTURNA addresses. And then finally, the clinical trial for LUXTURNA got off the ground at Children's Hospital of Philadelphia in late 2007. And the results from even the early stage trial were quite dramatic, as you heard Jake talking about. People, kids, young adults putting away their canes, seeing the faces of loved ones. Some folks even saw stars in the sky for the first time. And so ultimately, this gene therapy was approved in the US by our FDA in late 2017. We're coming up on the 4-year anniversary. And I think one thing that's really important to keep in mind about this treatment, because it was the first FDA-approved gene therapy for the eye or an inherited condition, it provided affirmation to the research community and the biotech community that gene therapy was indeed an effective way, in many cases, to address inherited retinal diseases. And now you see just a real explosion of activity in gene therapies for retinal degenerations. And as I'll talk about in a moment, some of these gene therapies are gene agnostic. That might sound like a contradiction, but you'll understand what I mean as I get to those examples. So some other gene therapy trials that are very exciting right now include those for a gene called RPGR, which when mutated causes X-linked retinitis pigmentosa. And there are two companies, AGTC and MiraGTx with their partner Janssen, who are in or moving toward phase 3 trials for RPGR gene therapy. Both of these emerging therapies in the earlier trials showed some impressive efficacy. And if they are successful in phase 3, I'm sure these companies will pursue regulatory approval. Now, one company I don't have listed here is Biogen. They completed their phase 3 trial for their X-linked RP gene therapy. Unfortunately, they did not meet their endpoints, so they've stopped clinical development. And that's just a sobering reality of clinical trials that we need to always keep in mind, that a lot of clinical trials fail. It's just the nature of science, and that's why we need so many clinical trials in the pipeline. And so another company that is addressing RPGR is called 4D Molecular Therapeutics, or 4DMT. Their approach is a little different. The AGTC and Mira are injecting the vector underneath the retina. It's a relatively invasive surgery. 4DMT, they are injecting the therapy into the vitreous, into the gel, the middle of the eye, much less invasive. The challenge there is getting the therapy to close to the retina, to the cells that need it. But if they can do that, an intravitreal injection is potentially a safer approach. So lots of great gene therapies in the pipeline for X-linked RP. Now, another company I want to talk about is called Atsena Therapeutics. This is a company that we've invested in through our venture philanthropy fund, and it was co-founded by Shannon Boy out of the University of Florida. And they have a few different therapies in the pipeline. One is in a phase 1/2 clinical trial. It's gene replacement for the gene GUCY2D, which when mutated causes Leber congenital amaurosis. Just a few months after this trial started, they reported some encouraging results for the first 3 patients, even at a low dose. So we're very encouraged by that. And hopefully, as the trial moves on, they'll continue to see efficacy and maybe even greater vision improvement as they move to higher doses in more patients. Now, Dr. Boy is also developing a couple of other gene therapies not in the clinic yet. One is for the gene Myo7A, which when mutated causes Usher syndrome type 1B. You heard Dr. Stevenson talking about some genes are too big for the viral delivery systems. Myo7A is one of those. So Dr. Boy is coming up with a technology where you deliver the gene in two pieces or two halves. And when those halves get to the retina, they combine so you get a complete gene and hopefully a functional protein. So she's moving that toward the clinic. And they recently also announced gene therapy development for X-linked retinoschisis, which is a pretty challenging and aggressive retinal disease. So another company that has a lot of great activity in the gene therapy space for inherited retinal conditions is Iveric Bio. I've listed 5 projects here. None of these are in the clinic yet, but they're all moving toward clinical trials. The last 3 on this list, which are gene replacements for CEP290, which when mutated causes LCA, ABCA4, which is the culprit usually when mutated for Stargardt disease, and also USH2A, which when mutated can cause Usher syndrome type 2A, and also non-syndromic RP. Those again are those large genes that don't fit into the vectors, into the containers we use to get the genes into the retinal cells. But what Iveric is doing is they're collaborating with research centers that are shrinking these genes so they can fit into the viral containers. Iveric, their most advanced project is for Best disease, which is a form of inherited macular degeneration. That gene therapy is getting close to moving into a clinical trial. And they're also developing a gene therapy for rhodopsin, which when mutated causes dominant RP. That's a pretty common gene associated with RP. Rhodopsin, because it leads to the production of a toxic protein, you need 2 steps. Not only do you deliver a new gene, but you have to shut down one of the copies of the mutated gene so that you stop production of that toxic protein. So I also wanted to mention a very exciting development for us at the Foundation Fighting Blindness. So we fund researchers and companies all over the world, but recently we announced the formation of our own company. This is the Foundation's own privately held company called Opus Genetics. It's been established with $19 million in initial seed funding, and we're specifically targeting some of the more rare retinal conditions, especially forms of LCA, and these will generally be gene therapies. So our first 2 targets are LCA5, which is caused by mutations in the gene laborcillin, and LCA13, which is caused by mutations in RDH12. And our scientific collaborators are out of the University of Pennsylvania, including Dr. Gene Bennett, who was the visionary for LUXTURNA. So we're very excited to have our own company and to be addressing these really rare forms of retinal degeneration. So up until this point, everything I've been talking about is what we call gene replacement. The scientists prefer to call it gene augmentation. The bottom line is you're delivering a new gene to replace the mutated gene. You're delivering many copies of that gene to replace the mutated copies. This form of genetic therapy is a little different. And you heard before from Dr. Whelan about RNA, DNA and RNA. There's a messaging system in our cells that carries genetic information through the cell so that the cell can make the protein that's needed for the cell to function properly and be healthy. So RNA are the genetic messages. So you can replace the gene, the DNA, or you can try to correct the RNA. And this company out of the Netherlands, ProQR, is targeting the RNA. And they've come up with this technology. It's got a very long name. It's called an antisense oligonucleotide. But what it is, it's just a tiny piece of genetic material that covers up the mutated region in the RNA. I kind of think of it as like a mask or like Velcro. And in the bottom right here, in this diagram I have on this slide, you can see this red bar covering up a piece of the RNA, almost like a piece of Velcro. So anyway, ProQR is targeting 3 different common mutations or regions that cause retinal degeneration. A specific mutation in CEP290, a region, a hotspot in the gene USH2A, and also the P23H mutation in rhodopsin. Now, the CEP290 and USH2A therapies have worked well in early stage clinical trials, and the CEP290 is now in a phase 2/3, and the USH2A therapy is moving toward phase 2/3. So we're very excited with the encouraging results coming from those 2 trials. The rhodopsin trial started a little later, and we're hoping to hear results from that study sometime early next year. So we're going to now discuss another variation on the form on genetic therapies. And again, this approach is not like gene replacement. In fact, it's gene agnostic. What you're doing in this particular project, which has been developed by Sparing Vision out of France, is you're delivering copies of a gene to the retina that express a protein that can help keep the retina healthy and slow down degeneration. And it's designed to specifically preserve cones. Cones give us central vision, visual acuity, color vision. They're really the cells that are most important for the vision we use for most of our daily activities. But the idea here is this therapy will preserve cones, and it will hopefully do so independent of the mutated gene causing the disease. So this is designed for people with RP, Usher syndrome, potentially other related conditions. And actually, I do need to update this slide. The phase 1/2 clinical trial is now planned for sometime in 2022. But for people for whom there isn't a gene replacement therapy, or for people who have these conditions and they don't know what their gene is, this approach is potentially relevant and promising because, again, it's designed to work independent of the mutated gene. So we're going to talk about yet another form of gene therapy, and this approach is for bestowing light sensitivity to a retina that has lost all its photoreceptors, and this is called optogenetics. And the particular project I'm highlighting here is called Bionic Sight, but there are other companies in this space. So on the bottom left portion of this slide, I have a side view image of a retina, and at the top I have this ominous looking X going through the photoreceptors, going through the rods and cones. As we've learned, rods and cones are the cells, the photoreceptors, that make vision possible. Well, in somebody who's lost all their rods and cones to an advanced retinal disease, all the other therapies I've talked about won't apply because they don't have any rods or cones left to save. But some very clever researchers observed that other cells in the retina survive after rods and cones are lost. These include ganglion cells and bipolar cells. And what they've done is they've developed a gene therapy that bestows light sensitivity to these bipolar and/or ganglion cells in the retina. These cells normally don't respond to light. They have other functions, but by delivering this gene to these cells, you can make them operate something like photoreceptors. So you can think of this approach as harnessing ganglion cells and bipolar cells for vision as like a backup system for somebody who's lost all their photoreceptors. What's nice about this approach, it is gene agnostic. It shouldn't matter in many cases what mutated gene is causing your disease. And this is for somebody who's really lost just about all or completely lost all of their vision. So Bionic Sight is one of the folks earlier this year that reported some rudimentary vision improvements in their phase 1/2 clinical trial. Two other companies, GenSight and Nanoscope, have also reported some early vision improvements in their early stage clinical trials. Now keep in mind, because we're not using photoreceptors for vision here, the visual experience of people receiving this treatment isn't going to be exactly the way we normally think of vision. But if you can provide even a rudimentary level of vision for somebody who's completely blind, that can be a big deal. So we're excited about this approach because it provides hope for people who, again, have lost all their vision, and it is gene independent. So I want to switch over to cell-based therapies and talk about a few different projects in that realm. So in this particular project, what researchers are using, and this is being developed by Renuron, a company actually in the UK who has a trial ongoing here in the US, but these cells that are being transplanted into the retina are called retinal progenitors. And you can think of them as like stem cells that have almost fully developed into photoreceptors. And the idea here is to inject these underneath the retina, and hopefully these cells will mature and actually connect with the host retina to restore vision to people who have lost photoreceptors. This is being evaluated in people with RP and Usher syndrome. In the phase 2 trial thus far, it's improved vision on average of about 3 lines on an eye chart. And that vision improvement has been durable for out to 12 months. And durability is obviously very important. Renuron is expanding this trial. And if they can continue to get some good data out of this study, they'll move it to a phase 3. Now, another stem cell trial that launched actually a little more recently than the Renuron trial is for people with advanced dry AMD, and we call that geographic atrophy. AMD, a complex disease, causes, in advanced forms, causes devastating central vision loss. But what's really cool about this trial is they're using stem cells that are derived from the skin or the blood of the patient. So what researchers can do, including this researcher from the National Eye Institute in the US, Kapil Bharti, is he can take the skin or blood of a patient, he can genetically modify those cells so they revert back to a stem cell state, and then he can coax those cells forward to become cells in the retina, maybe RPE cells. Well, that's actually what he's doing in this particular clinical trial. In people with age-related macular degeneration and Stargardt disease, the disease often manifests in a cell layer called the RPE cells. And when the RPE cells degenerate, the photoreceptors follow. So the idea here is to transplant these RPE cells derived ultimately from skin or blood of a patient and put them in the retina. And he's putting them on a biodegradable scaffold so they're better oriented in the retina when they're transplanted, and they can integrate and survive better. So this is phase 1/2, and we're just waiting to hear results from this early stage trial. Now, this particular project is not ready for the clinic yet, but it's a really cool project, and I really felt compelled to include it in this slide deck. So all the cell-based therapies that we've heard about today and the other ones that are in development involve taking cells from an outside source and putting them into the retina. Well, imagine if you could grow your own new photoreceptors. And that's exactly the approach that's being developed by Tom Ray at the University of Washington on the west coast of the US. So what Tom Ray observed in amphibians is when amphibians lose their tails or arms, or they lose retina, amphibians are able to grow those body parts back, including retina. And what he did is studied the factors that enable these creatures to regenerate their own tissues. And he's come up with a small molecule that he's used in mice and is now using in a large animal model to enable these higher level species to regenerate their own retinas. Because outside of amphibians or some reptiles, mammals aren't able to regenerate their own retina. So what would be really great about this approach, if he can get it to work in mammals, is that you don't have to transplant anything. He can get photoreceptors to sprout from another layer of cells called Mueller glia. So we're very excited about this, and Dr. Ray has made good progress, and we're working to help him move this into a clinical trial. And so finally, to start rounding things out, I wanted to talk a bit about therapies for Stargardt disease. And there are actually a number of clinical trials underway to try to slow the progression of Stargardt disease. And it's important to understand why Stargardt disease occurs. And the simple explanation is we all need vitamin A to see. And most of us in developed countries get plenty of vitamin A from a normal diet. Even if you don't eat a lot of fruits and vegetables, you're going to get enough vitamin A in your diet so that your retinas can be light sensitive. But if you think of vitamin A as being like a fuel, when vitamin A is metabolized in the retina, there are these byproducts, these waste products that are produced that need to be cleared from the retina. And in people with Stargardt disease, the problem is those waste products are not cleared. They accumulate in RPE cells, and that leads to degeneration. So these 4 companies, or at least 3 of these on this slide, are targeting that vitamin A waste pathway. So Alkeus is a company that has developed a form of vitamin A that, when metabolized, doesn't lead to these byproducts. So people take this modified form of vitamin A, at least they have in a clinical trial, so that when that's processed, they don't have these harmful products. So Alkeus is the company that's doing this. They completed their phase 2. Informally, they actually had some pretty good results, and now they're working toward a phase 3. They haven't formally published their results, and we really want to see those before we get too overly excited about what's happening there. But informally, it sounds like they've had some encouraging outcomes from their trial. Companies Acucella and Stargazer are working on approaches that minimize the uptake or the processing of vitamin A in the retina, because if you can slow down that process or inhibit that process a little bit, you can slow down the accumulation of toxic byproducts. And then finally, Iveric Bio, who we heard about, is actually targeting a different pathway. They're targeting what we call the complement system, which is a part of our immune system. And what Iveric believes is if you can target the overactive immune system in people with Stargardt disease and potentially other conditions, you can slow down the degeneration, because it seems as though in AMD and potentially other inherited retinal diseases, it's this innate immune system that's overactive that's a major problem in retinal degeneration. And then finally, I wanted to say that for Stargardt disease, we do not currently have a gene therapy in a clinical trial, but there are several companies that are working in preclinical studies to move their gene therapies, their ABCA4 gene therapies, into the clinic. And they include AGTC, a company in Europe called Cove, and Iveric Bio. Again, ABCA4 is a big gene. You either have to shrink it or deliver it in 2 pieces, so it makes it a little more challenging. But I think it's heartening to know that these efforts are underway. And again, they are moving toward the clinic. So finally, to close things out, I wanted to mention that if you want to learn more about these projects and a lot of other research underway, you can go to our website, fightingblindness.org. We have a patient registry that's open to anybody in the world. You can get on the radar screen of clinical trial recruiters by being in that registry. And then clinicaltrials.gov is a great site to get information on clinical trials. And the picture I have on the left here of donuts was taken at a shop right outside of Stephen's Green in Dublin when I was there 3 years ago, called the Rolling Donut. And that was a favorite stop of mine every morning. Excellent, excellent donuts. That's an important part of the journey, is making sure you get good donuts. So anyway, that concludes my talk. I hope I didn't go too far over my time, and I'm not sure if we have time for questions or not.
Thank you very much, Ben. I think we've had a little bit of time. Thank you very much. That was very informative. I'm going to ask them as they came in. I'm not a scientist. Do you know anything about the PROM1 gene for cone rod dystrophy?
Right. I know a little bit about it. And I think some of the therapies that are targeting central vision and cone vision might apply to PROM1, but they're not at a stage yet where we've evaluated those in PROM1 patients. So stay tuned as some of these cone targeted therapies emerge to see if we can apply them to people with PROM1.
Thank you very much, Ben. Is
Sure.
anyone working on Stickler syndrome, do you know?
You know, that's a great question, and offhand, I honestly do not have the answer. What I would say is potentially some of these gene agnostic therapies might apply, but if this person wants to email me directly, bshaberman@fightingblindness.org, I'll type that into the chat here. I can look into it, but I'm not sure offhand.
Okay, thank you very much. And the final question, the BBS1 gene. Are you familiar with the BBS1 gene?
Sure.
Is there a gene therapy emerging with that in mind?
Sure. So BBS1, when it's mutated, I know it can cause Bardet-Biedl syndrome. That's where it gets its name. And I think potentially it can also cause non-syndromic RP as well. So the best way to think of that particular gene is like an RP gene, at least from a retinal standpoint. And the gene agnostic approaches that I mentioned, like optogenetics, sparing vision, some of the cell-based therapies, those might apply to people who have mutations in BBS1. Because again, you can think of it as just like RP from a retinal standpoint. But one thing I want to mention about Bardet-Biedl syndrome, if this is what this particular person or their loved one is affected by, it can cause myriad of health issues beyond retinitis pigmentosa. And we're actually talking to a company in the US called Rhythm Pharmaceuticals, who is developing a drug to help manage the obesity that can be so common in people with Bardet-Biedl syndrome. So I think that's an exciting development. They're working with FDA to try to get approval for a drug to better manage that obesity. So again, that's Rhythm Pharmaceuticals, and they're in the Boston area.
Okay, thank you, Ben. Time for one more, I think. What about where are we for full transplant? Where's the research for full transplant capability?
So that's always a good, intriguing question. There's a lot of challenges to replacing the entire eye. One of the challenges is the optic nerve, which connects the eye to the brain. That's a cable with 1 million fibers in it, and to make that connection work well is pretty challenging. You know, there are muscles, there are other things that need to connect the whole eye into the socket of the recipient. So we're still a ways from accomplishing that. The good news is people are studying that. At the University of Pittsburgh, I know there's a researcher, Keo Washington, if I remember her name correctly, who is working on that challenge. Again, not ready for prime time. We got a ways to go. I would say if you can do more with the retina itself or harness other cells in the retina for vision, that's a nearer term solution to somebody who's lost a lot of vision. Finally, I'll say there are a couple of companies that are developing devices that connect directly to the visual cortex, the part of your brain that makes vision possible, so that you can bypass your ocular system entirely for vision. Now that's still kind of science fiction. It's in early stage clinical trials, but that is another pretty interesting approach that people are taking to restore vision.
Ben, thank you so much for bringing us all up to date with the latest advances in retinal research,
My pleasure.
which, as we've established, is a bedrock for future treatments and cures. So thank you so much, and we really hope to see Ben in Ireland again soon, perhaps next year.